Okay. Good morning, everybody. Can you all hear me well? Yes? Excellent. So it's a true pleasure for the team Interdisciplinary Blood Science to welcome Laurent Yvernogeau today. For those who do not know Laurent, he's been trained as an embryologist and a developmental biologist from the very beginning of his career during his PhD in Nantes in the lab of Josiane Fontaine-Perrus, where he started to work on the endothelium and the smooth muscle development in the mouse. And then he moved to Thierry Jaffredo's lab for a postdoc in Jussieu in Paris, which is a world-class lab in hematopoietic stem cells and the development of the hematopoietic system. So there he switched to developmental hematopoiesis, and he had seminal contribution to the field where he developed the first ex vivo culture system allowing to recapitulate the very early events, very early steps leading to the generation of the hematopoietic stem cells. And he also challenged the dogma of basically the main idea that hematopoietic stem cells are only formed in the embryo during at mid-gestation, but he showed that actually this dogma is not entirely true and that you can generate de novo stem cells from the bone marrow after birth, so outside the embryonic wave. That was published in a Nature Cell Biology paper a couple of years ago. And then he continued his career as he did another postdoc in the lab of Catherine Robin in the Ubrecht Institute in Utrecht in the Netherlands. And Catherine is an expert in the analysis of the emergence of the very first stem cells in the embryo, hematopoietic stem cells. And there he performed an outstanding work, basically identifying the molecular components and interplay of the different signals that contribute to hematopoietic stem cell emergence, which has a far-reaching potential for regenerative medicine and basic understanding of this complex tissue. And now he's back in Paris, in the Institute of the IBPS in Paris, with Thierry Jaffredo, and now he's a PI there and started his own lab, continuing to work on that topic, but using very original approaches, using different developmental systems, the quail, the chick embryo, the mouse, and the human embryos. So together with cutting edge technology. So Laurent, welcome. We'll be very happy to share your stories. Feel free to start whenever.
Okay, can you hear me? Fine?
I can hear you well.
Okay.
Just so, before you start, if during the talk people have questions, feel free to write them on the chat. Or if you have a burning question or if something isn't clear, feel free to light up your camera and interrupt Laurent, and we'll be happy to discuss in a live setting. Okay, let's go.
Okay. So thank you very much, Eric, for the very nice introduction, very kind introduction. Can you see my screen? Eric, are you there?
Sorry, my microphone was switched off. Yes, I can see your screen.
Okay, perfect. So I will start. So thank you again, Eric, for the invitation and for this very nice introduction. So today it's really a great pleasure to give you a seminar on the topic I'm working on. So I will talk about hematopoietic stem cell emergence, and basically I will present you the work I've been doing in the past, as Eric's mentioned, when I was at the Ubrecht Institute. So that will be more or less the first part of my talk. And the second part, I would like to share with you the projects that I'm now currently developing here. So these are totally new and unpublished data. But let's start. So a quick introduction on hematopoietic stem cells. So these cells, as you might know, are located within the bone marrow in the adult. They are characterized as being multipotent and self-renewable. So self-renewable meaning that they can maintain the pool of hematopoietic stem cells during the whole life, and they can differentiate in all different blood cell types. And this is basically the definition I will use. So to define an HAC is a cell able to reconstitute the hematopoietic system of an irradiated adult mice at long term. So these hematopoietic stem cells are responsible for the blood cell production. They are really important in clinic because we use these cells to treat patients with blood-related diseases. And even if we made a lot of recent advances, it still remains really challenging to isolate these cells, to maintain these cells, or to expand these cells in culture. And this is probably because we still don't really know the full steps that lead to the generation of these hematopoietic stem cells. And even if these cells are located within the bone marrow, they are actually generating really early during embryonic development. So in the next few slides, I would like to, I will take the mouse embryo as an example, but the basic rules are the same for all vertebrates. But are these hematopoietic stem cells arrive during development? So if we took the mouse embryo, it's around mid-gestation at E10.5 that you detect the first hematopoietic stem cell activity within the aorta of the embryo. And on transversal section, it looks like this. So you have the big vessel, the aorta in the middle, and surrounding by the gonad and mesonephros. So we call this region aorta, gonad, mesonephros, and it is within this region that you can detect the HAC activity. Soon after, this HAC number increase a bit, and they are detected also in the main arteries, like the vitelline artery or the umbilical artery, and starting around, and also in the placenta, sorry, and starting around E12, this HAC move and are found within the fetal liver. And within this fetal liver, they are gonna be massively amplified. So the fetal liver really is a site where HAC are expanded. And starting around E17, a bit before birth, these HAC have to leave the fetal liver and colonize the bone marrow, where they will reside for the rest of the life. So this is really quite a journey that this hematopoietic stem cell has to undergo, and they travel to different sites before to reach their final destination. But still, within this aortic region, the question remains where this HAC arise. And it was observed a long time ago, like a century ago. If you do transversal section of an embryo, and you look here at the aorta, this is a mouse aorta, it was observed this group of hematopoietic cells tightly attached to the endothelium of the aorta. And we call these cells intra-aortic hematopoietic clusters. They are observed in all species that were studied, like in the human, in the avian, the xenopus, and this is among others. And it's really a common feature of all vertebrates. So it was proposed that this group of hematopoietic cells contain the first HACs, but where exactly and how these intra-aortic hematopoietic clusters are generated. Because this hematopoietic cluster cells shared some common marker with the endothelium of the aorta, it was quickly proposed that this group of hematopoietic cells are deriving from the endothelium of the aorta. And the first proof was done in the avian system. This was done by Thierry Jaffredo. What he did was basically to inject within the embryo a dye that can be incorporated within the endothelial cells. And he did this injection before the emergence of hematopoietic cells. And when he analyzed the embryo 1 day later, he realized that these hematopoietic cells that are attached to the endothelium retain the dye, showing that they come from, indeed, from the endothelium of the aorta. The same observation was done later on the zebrafish model using transgenic line like this KDRL GFP. So basically in green, you see the endothelial cells. The aorta is the upper vessel and the vein is underneath. And using this transgenic line combined with time-lapse imaging, the group of Philippe Herbomel and Karima Kissa demonstrated that some endothelial cells of the aorta at some point change their shape. They become more round, and they detach from the endothelium, go in between the space between the aorta and the vein, and at some point they will join the circulation via the vein. And it was really shown that these endothelial cells that are detaching from the aorta are becoming hematopoietic. At the same time, same observation done in the mouse. This was done in the lab of Catherine Robin, where they did thick transversal section of the mouse embryo. They stained the endothelium of the aorta with antibody like CD31, and they performed time-lapse imaging. And during this time-lapse imaging, they observed some cells budding in the lumen of the aorta, expressing hematopoietic marker like SCAR1. So the same process occurring in the mouse. And an important observation, if you analyze some mice which are lacking the expression of ROX1, you do not form this group of hematopoietic cells, you never generate HAZ, and these embryo die really soon. So ROX1 is definitely a critical player in this process. And it's now commonly admitted that basically endothelial cells differentiate from the mesoderm. Some of endothelial cells of the aorta become what we call hemogenic endothelial cells that undergo what is called the endothelial to hematopoietic transition, where ROX1 is critical to generate the first hematopoietic stem cell. Now on this slide, I put like a summary of what is known on the 4 classical models used to study developmental hematopoiesis. And so basically in the zebrafish, it's around 40 hours post-fertilization that you have the maximum emergence of these hematopoietic cells. In the middle part of the embryo and in this aorta, the cell emerge as single cells that are immediately released through the circulation. In the mouse, it's around male gestation that this process is occurring, is maximum mostly concentrated in the middle region of the aorta. And on section, this is occurring mainly in the ventral side of the aorta and to a less extent also to the dorsal side. In the avian system, it's at 3-day post-incubation that you have this maximum emergence of hematopoietic cells within the anterior part of the embryo. And on section, this is occurring in the ventral side only, and it's the full floor of the aorta that is becoming hemogenic and producing these hematopoietic cells. And in the human, it's at 30 days post-fertilization that you have this emergence in the medial part of the embryo. And as in the chicken, it's occurring only in the ventral side. So this is to show that the process is really conserved in between species with few discrepancies. But we do believe that if this process is occurring in specific region and its specific time point during development, it's probably because of the surrounding microenvironment of the aorta that is producing signals, allowing this emergence to occur in this specific location at this specific time point. And this was the question I wanted to ask when I was postdoc at the Hubert Institute. And I wanted, because I was coming from a chicken lab, I wanted to use the chicken embryo as a model. But before to go a bit more in details, I thought at that time that I still missed some important information in the avian system, like I don't really know how many cells are generating within the aorta of the chicken, how they are really organized along the embryo, and also in time and space. So to answer this first question, I wanted to make a very nice cartography of this hematopoietic cluster emergence within the chicken embryo. And for that, I did a lot of Woolman staining, which basically, so you perform immunostaining on the full embryo. So on this movie, what you will see is the blood vessel in red, the hematopoietic cells are in green, and in blue, it's the ROX1 expressing cells, so the cells that are doing this endothelial to hematopoietic transition. And by doing this Woolman staining, you can really have a nice view of the organization in space, because you can do like this one, a 3D reconstruction. You can really zoom in some specific area. Now we are zooming in the anterior part of the embryo, where we can really see that the blue cells, or the hemogenic endothelial cells, are really present in the ventral side of the aorta. And it's really concerning the full floor of the aorta that is at some point becoming hemogenic and producing these hematopoietic cells. Then, of course, you can zoom up, and we can zoom more to the posterior part of the embryo. And you will see that basically after the vitelline artery connection, after this part, you don't find any blue cells anymore. So this hematopoietic emergence is occurring really in the anterior portion. So using this Woolman staining, you can really have a nice 3D reconstruction and visualize how the cells are organized. And what you can actually do is really zoom in deep within the aorta, as it's shown here, and you can actually, going through the Z-stack, count every single cell that are generating in the aorta. And this is what I did for all the developmental time point of the chicken embryo. So now we have a nice idea of how many cells are generating within the development of the embryo. And what we also noticed during this, project is that actually the hematopoiesis in the aorta of the chicken is occurring as a wave, progressing from the anterior part to the posterior part. I mean, at early time points, this emergence is really concentrated to the most anterior part of the aorta, then it's shifting to the posterior part, and at later stage point, even posterior, but never after the vitelline artery connection. So it's really a wave of emergence along the anterior posterior axis. And now I was really feeling confident that this is a very nice model to tackle the question why this is occurring in specific areas like in the anterior portion only, and why it is occurring only in the ventral side only. And obviously, it's because of the surrounding tissue of this aorta that probably inhibit this emergence to occur in the dorsal portion, while at the other hand, in the ventral part, in the ventral mesenchyme, there must be something allowing this emergence to occur. But how can we identify the gene expressed in these specific areas? So basically, what is the role of the microenvironment in this emergence? To identify the gene expressed in this specific location, I used the technique that is called TomoSeq. TomoSeq, it was developed at the Hubert Institute, and I will explain now the principle of the technique on this few slides. Basically, the idea is to use a whole tissue. So this is again the chicken embryo. The posterior part is here, the anterior part is here. I remove the head to get a bit less tissue, but this is the heart of the embryo. And the idea of the TomoSeq is to perform cryosection of this unfixed tissue according a defined axis. So here I choose the posterior to the anterior axis. Every section is collected into one Eppendorf tube. Then you extract the RNA for all the tubes and you sequence everything. And after normalization, what you can obtain, you can look for the gene expression along the axis that you choose during the cryosectioning. I'm showing you here the expression of NKX 2.5, which I know it's expressed specifically in the heart. And you can see on the TomoSeq data that you have only expression of this NKX on the section that were containing the heart. So you really have a spatial resolution along the embryo axis. So then I use this TomoSeq to identify genes expressed in specific areas to answer my question. So again, I use chicken embryo. I dissected the aorta from its E3 embryo, so the full length of the aorta. This is the posterior part. The anterior part is here with blood cells in the aorta, keeping, of course, the surrounding tissue, because this is where I want to identify genes. I also apply this TomoSeq on 6 transversal sections that I recover from the anterior portion. It looks like this, because I know there is emergence of hematopoietic cells in this ventral part. And to have some contrast, I also use 6 transversal sections from the posterior side, which is looking like this, because I know that here there is no emergence of hematopoietic cells. And we apply the TomoSeq on all these samples. So this is what we can get. Here I'm showing you the expression of MPL, because we know MPL being expressed by the hematopoietic cells that are generating within the aorta. And you can see that on this sample, we have no expression on the posterior part and the peak of expression in the anterior portion of the aorta where emergence of hematopoietic cell is occurring. It's even more clear on this section. You have only this peak of expression of MPL that fit perfectly with the ventral side of the aorta where there is emergence of hematopoietic cells. And in the posterior slice, no expression. And the technique is really powerful because now you can define, as I did, define some region of interest and also ask to look for all the genes that follow the same expression pattern as MPL. These are the gray traces that you can see that I call xmRNA. And you can start to dig into this data to select and identify potential new candidate genes that can be responsible for the emergence of the hematopoietic cells in this specific region. But the problem with this approach only, it's not only you will identify genes that belong to the hematopoietic cluster cells that are generating, but also some that are specifically expressed by the microenvironment. So to discriminate between these two possibilities, what I did was to FACS sort some hematopoietic cluster cells of the aorta and perform RNA sequencing on these hematopoietic cluster cells. So for that, what I did was to inject directly in the aorta antibodies that can stay the endothelium of the aorta or only the hematopoietic cluster cells, and basically dissociate the tissue, go to the FACS and isolate only these hematopoietic cluster cells and perform RNA sequencing. And because I did that, now I know within my TomoSeq data which genes belongs to the hematopoietic cluster cells and which genes belongs to the microenvironment. Then, of course, you need to validate the genes that you identified, and we did that by in situ hybridization. So just to show you again that with the TomoSeq, you can identify genes expressed specifically by the hematopoietic cluster, MPL, as I just mentioned, expressed specifically by the cluster. You can identify potent new genes like LY86, also expressed specifically by the hematopoietic cluster cells. And you can also, with the TomoSeq, identify gene expressed by the microenvironment, like LSP1, which is nicely expressed just underneath the ventral side of the aorta, or this phosphatase protein, which is expressed by the gonad. So the technique was really validated using the chicken model, but of course I wanted to have a species comparison. So I basically applied the TomoSeq on other vertebrates. And what I did, I applied it on 4 different species on human samples, 2 different samples that we managed to obtain at in the mouse embryo at different time points, and in the zebrafish embryo also at different time points and different axes. And we applied this TomoSeq on all these samples. So that was quite an effort. I think it was like 28 sample process for the TomoSeq. And with that, what we did, we, as I mentioned before, we defined some region of interest. So we basically defined some region where we want to identify genes more expressed in the red area compared to the rest. And we applied this filtering strategy for all of the species. We then obtained a list of candidate genes for all the species, and we compared the genes list together. So this is the Venn diagram that we obtained at the end. I have to mention, so this is only 3 genes in common to the 4 species, so it might look very few, but at least these 3 are known to be important in developmental hematopoiesis. And I want to mention that this is low because of the filtering strategy that we applied. We define some strict region of interest, we apply some strict threshold, but of course, if you increase the region of interest, if you decrease the threshold, you will increase this list. So of course, and we find a lot of genes that were already known to be implicating in leukemia or upregulated in a supportive stromal cell line. And we, yes, of course, obtain also a list of candidate, new candidate genes. What we also did as an analysis, we wanted to look for the possible crosstalk between these hematopoietic cells and the microenvironment. So we look for ligand receptor interaction, and we can do that because we sorted some hematopoietic cells from the chicken, and we also have the data for the mouse. So we basically look what ligands we find within the TomoSeq in the microenvironment and the receptor present in the cluster, and how these can be, how can these two can crosstalk. So this slide is a bit busy, but I kind of like it because I think it really reflects the complexity of this microenvironment during this emergence of hematopoietic cells. And among these, we picked 2 that were not really defined as regulator of HSC emergence within the aorta, which is ADM and ROM2. ADM that we validated, the peptide is expressed really ventrally, more ventrally in the aorta, and ROM2 being expressed by endothelial cells and hematopoietic cluster cells. So we picked these 2 to push a bit further the validation, and we perform functional validation of these genes. Another one that we validated, which is not here, it's VEP1. VEP1 is a secreted protein, so I wanted to validate different kind of candidate. So Let's start with IDM ROM2. So adrenomedulin and its receptor are known to be a potential and vasodilatator agent. And at that time, we thought it might be really a good candidate since we know that hemodynamic shear stress is really important for hematopoiesis to occur within the aorta. This was really known. And we thought maybe this is a sensor of this hemodynamic shear stress. So that's why we picked this candidate. And to validate its function, we perform knockdown experiment. So we basically knock down adrenomedulin and ROM2 by injecting morpholino at one cell stage in the zebrafish embryo, let the embryo grow until they reach 40 hours post-fertilization, and then perform in situ realization for RONX1 to see if this absence of IDM and ROM2 disturb the generation of hematopoietic cells. We also perform validation on the chicken model. And what I did was to inject in the heart the adrenomedulin peptide in the chicken embryo before hematopoiesis in the aorta, let the embryo grow, and evaluate the consequences on the hematopoiesis by flow cytometry. So for the morpholino experiments, so this is the control morpholino, and this is the in situ for RONX1 as it can in the control fish. So what you see is dot of blue cells, so basically cells expressing RONX1 emerging along the aorta. And if you knock down either IDM or ROM2 with two different kinds of blocking morpholino, you can appreciate that you really decrease this number of blue cells. So you decrease hematopoiesis in the aorta. And you can actually count these cells. And this is what we did. And you can see that with ADM or ROM2, all the time you will decrease this number of hematopoietic cells emerging in the fish, showing that adrenomedulin and ROM2 is crucial for this emergence. So in the chicken, what I did was to inject the agonist form of adrenomedulin or the antagonist and perform flow cytometry. And if we inject the agonist form of adrenomedulin, we increase the amount of hematopoietic cells generating within the aorta, while the antagonist seems to decrease this production. So this was for adrenomedulin and ROM2, but we also wanted to validate another type of candidate like SVEP1, a secreted protein. So he has this very long name, Sushi von Willebrand factor type A, EGF, and pentraxin domain containing 1. He has a well-defined function for lymphoangiogenesis. The fish and the mouse who lack the expression of SVEP1 develop huge edema, and this is really lethal for the fish and the mouse. And recently, it was also shown that SVEP1 is important to maintain HSPC on stromal cell line from the spleen. So we chose to validate SVEP1 at the time because we get the fish and the mouse knockout line. And we're not going to show the result for the fish because we did more or less the same experiment as I just showed for adrenomedulin and ROM2. But with the mouse, we performed transplantation experiments. So what we did was to use either wild-type embryo or knockout embryo at E11, where we dissected the aortic region, and we injected this aortic region within adult recipient mice that were irradiated. And because the donor cells and the recipient don't have the same allele for CD45, and we do have antibodies specific for these two, what we can do after 4 months after the transplantation, we can bleed the mouse and check if we find hematopoietic cells coming from the recipient or hematopoietic cells coming from the donor. So basically, we can appreciate if the cells that we injected are able to repopulate the hematopoietic system of the recipient. And then if the percentage is above 5%, the mouse is considered as positively reconstituted. And we can look within this population if the cells we injected were able to differentiate into all the different lineages, showing that you are dealing with hematopoietic stem cells. So this is what we did. And these are the results. So basically, when we inject wild-type embryo, we have this amount of mice reconstituted and a bit less mice reconstituted with the knockout cells. Then we check within these positive mice whether we had all the different lineages, and this is the wild type, this is the knockout, and yes, we actually do have multipotential for all of them. So meaning that ZEB1, in the absence of ZEB1, the embryo do develop some multipotent HSCs. But since we had this small decrease, we were wondering what happened if we inject a bit less cells to reconstitute the mouse. So that's what we did. We injected a third of the cells, and then you get less mass reconstituted even with the wild type, but even less with the knockout. And because we did these 2 doses, we can then calculate the frequency of HSC that we are dealing by using the Poisson statistic law. And this is what we did, and we obtained 2 HSC per wild type embryo, which is what you find in the literature, but half less with the knockout ZEB1, showing that ZEB1 is actually indeed to get this normal amount of HSC. And unfortunately, we didn't get far with this, so we still don't really know how ZEB1 can regulate the generation of the RTR. This will need more studies. So just to summarize what I just showed you, so we basically apply this tomosic approach to have a special transcriptomic approach on 4 species: the human, the mouse, the chicken, and the zebrafish. With this technique, really powerful, you can identify genes specifically expressed by the hematopoietic cells emerging within the aorta, but also find genes expressed in the macroenvironment. This was published 2 years ago. And because we did so many samples, we also developed a website where all these samples are listed, and people can have a look. And I think it's a very nice resource, which looks like this, basically. So you basically enter your the name of your favorite genes here, and you can look then its expression profile in the chicken, the zebrafish, the mouse, or the human. And if you pick the zebrafish, then you always have a scheme of the samples and the way we process the sample. Then you get immediately the profile along the axis. It gives you immediately the genes following the same expression pattern. And the bit on the bottom, you have a list that you can download of all the genes following the same expression pattern of your candidate gene. So I hope this website will be really useful for the community. And all this data that I just presented to you was done when I was a postdoc at the Hubert in the lab of Catherine Robin. So I would like to thank all the people that were collaborating with the project and all the people that were helping me with this project.